Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR Net Life Science JRF question paper for the conducted in the year March 2025. This is of shift 2 question paper, and I will be discussing the part C section, and in this video I will be discussing mainly the genetic questions what they have asked in the part C. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, select for CSIR Net JRF exams, and there you will find. So coming to the first question. Stutrivant introduced the concept of recombination frequency based genetic maps. Yes. That is correct. You all know that he constructed the first genetic map of the chromosome, and he proposed that the frequency of recombination between the genes on a chromosome, and it is proportional to the distance which are separating these genes. Okay, and based on this he developed the genetic map. Then since then, genetic map distances between genes may be underestimated due to the occurrence of double crossovers. Other mapping functions were developed to provide a more accurate estimate. Okay, now what does this statement means is, now Stutrivant he made the simple map, genetic map. That is, he assumed that there is complete interference. Interference means if one crossover is taking place in one place, it will interfere with the occurrence of a second crossover nearby. So complete interference means no crossover occurring at all in a nearby place. So he was considering in general only a single crossover events, not double crossover events, or I can say not multiple crossover events. So what does the statement mean? That genetic distance between genes may be underestimated due to the occurrence of double crossovers. Now see this condition, A, B, C, and oh, this is small a. Okay, I made a mistake there. This is small a, small b, small c. Now upon crossing over, this has exchanged place. That is, the B has exchanged place. You get capital A, small b, capital C, small a, capital B, small c. Now again a crossover is taking place in the same region, and you will get the parental combination itself. Now will you be able to calculate the recombination frequency? No. As per you, they are looking the same, just like your parents. Okay, so there you underestimate that recombination has not taken place. So the and you underestimate the genetic distance. So such kind of double crossovers will lead to underestimation of the genetic distance between genes. Okay. So to provide accurate estimate, two other functions were developed, that is Haldane's mapping function, which assumes no interference. Just like the Stutrivant simple mapping technique, Haldane also assumed that there is no interference between crossovers, while Kosambi's mapping function accounts for interference. The following graph represents the relationship between recombination frequencies and map distance as proposed by Stutrivant and other corrections using either Haldane or Kosambi's mapping functions. Okay. Now, so this first one, it will be Haldane mapping function. This is the graph for that. This is the Kosambi's, and this is the simple mapping function. Okay. So simple mapping function, there is no double crossovers and there is complete interference, and it is a linear relationship. The graph is a straight line. That is linear relationship between recombination frequency and map distance. Okay. So greater distance between the genes, more is the recombination frequency. But this type of mapping doesn't take into consideration the double crossovers, triple crossovers, or I can say multiple crossovers, and this has led to underestimation of the genetic distance between genes. So that is the simple mapping function. Now coming to the Haldane one, that is the first one. So what did Haldane do? He took the multiple crossovers into account, but he didn't take the crossover interference into consideration. Okay. So no interference was assumed by Haldane. So because of this, Haldane, for the lesser map distances, there was proportionality between the recombination frequencies. But as the distance between the gene increased, recombination frequency will increase. And you can see multiple crossovers also taking place. Now because he didn't take interference into consideration, he got a deviation from the straight line like this. Okay, it became curved. That is what Haldane did. Now the second was Kosambi's. Here also, as the map distance increases, recombination frequency increases, and you got a linear line. But after certain distance, as the recombination frequency increases, the genetic map distance increases. You can see multiple crossovers and there is interference also. So Kosambi, he took into consideration interference along with these multiple crossovers was taken into consideration. Because of that, his deviation was lesser than that of Kosambi's. So second is lesser than Haldane. So Kosambi's is 2. Okay. Now line 1 in the above plot represents relationship between recombination frequency and map distance based on Kosambi. No, that is not correct. It is Haldane. So A is not correct. So first one is false. Oh, great. That means 1 gone, 2 gone, 3 gone. Have one first option. If you know the answer, then you can happily tick 4. Then next one. Then the chance of underestimation of map distance increases with increase in recombination frequency. Yes, as recombination frequency increases, double crossovers will take place as I have mentioned here, and it will result in underestimation of the genetic map distance. So B is correct. Then genetic mapping proposed by Shrutivan assumes complete interference. Yes, that is, if crossing over is taking place at one region, then a near occurrence of a crossover nearby is not possible. It will be interfered. That is complete interference. So that is also correct. So that makes option 4 as the right answer. Then next one. Normal yeast cells grow at 42 degrees. Okay. 5 yeast haploid. Okay. They have taken the haploid strains, not diploid, with independent alleles of YFG1 having impaired cell growth at 42 degrees. That means they became temperature sensitive. That is, they were growing at 42 degrees. Now they can grow only at lower temperatures, not at 42 degrees or higher than 42 degrees. Were isolated and labeled as YFG1 TS1 to YFG1 TS5. So YFG TS1, TS2, TS3, TS4, TS5. A haploid YFG1 TS1 carrying a spontaneously generated mutation SUP1 at an independent locus. Okay. Now they have taken a YFGS1 strain carrying the mutation for YFG1, and they have found that it is carrying a spontaneously generated mutation, that is SUP1, at an independent locus, not in the same locus as this one. And they isolated and found that it is able to grow at 42 degrees Celsius. So because YFG1 was mutated, it was not able to grow at 42 degrees. Now when they generated this SUP1 mutant, they found that it was able to grow at 42 degrees Celsius. Using pairwise crossing, SUP1 was introduced into strain. So they introduced this SUP1 in S2, S3, S4, S5 and they found that these alleles are also able to grow at 42 degrees Celsius. Okay. Now based on this data, the following statements were made to describe the SUP1-YFG1 molecular genetic interaction. Now they have found that, or they thought that, these researchers, they thought that there was some interaction going on between the SUP1 and YFG1, and because of that the YFG1 mutants were able to grow at 42 degrees Celsius. Okay, now what are the assumptions? SUP1 codes for a protein which when overexpressed stabilized mutant YFG1 TS proteins. Okay, now SUP1, okay, it is a spontaneously generated mutation and it codes for a protein that is, it will overexpress the protein, and because of the overexpression of the protein, the function of this mutation will be reduced and it will be able to grow at 42 degrees Celsius. Could be, we cannot say no. Yes, that could be one reason. Then SUP1 codes for a protein that physically interacts with YFG1 protein. Okay, now YFG sorry, mutants will be producing one type of protein and SUP1 will be producing another type of protein. These two proteins, they will interact with each other and will result in a normal phenotype, that is the mutant YFGTS1 mutant will be able to grow at 42 degrees. Okay, that is also correct. Then SUP1 protein upregulates an alternate pathway. Okay, then SUP1 also is acting on another pathway and it is upregulating the production of proteins by that pathway and thereby inhibiting the YFG1 mutation and thereby allowing the cell to grow at 42 degrees Celsius is also correct. So A, B, C correct, that makes 1 gone, B gone, second also gone. Now we have to see whether D is also correct. What they have told is SUP1 is a nonsense suppressor that restores protein translation in cells carrying YFG1TS allele. Now what is a nonsense suppressor? That means if there is a nonsense mutation, the mutation will result in truncated protein production. Now nonsense suppressor, it will suppress the nonsense mutation and you will get a correct protein. So that is what a nonsense suppressor does. Now if SUP1 is a nonsense suppressor, then the YFG1 mutation has to be a nonsense mutation. Okay, if SUP1 is a suppressor, then the mutation caused by YFG1 has to be a nonsense mutation. Only then SUP1 can act as a nonsense suppressor there. Okay, but in the question they have not mentioned that it is a nonsense mutation. They have just told that it is a mutation. That's all. Okay, just mentioned that it is just a mutation. Now I will tell you why it is not a nonsense suppressor. So I told you that YFG1 has to be a nonsense mutation if SUP1 is to be a nonsense suppressor. Now if YFG1 is a nonsense mutation, then the nonsense mutation, they will result in the production of null alleles. Null allele means the resulting protein that is produced will be non-functional. Okay, or there will be complete loss of function of that particular allele. That is null allele. Then on top of that, they have mentioned in the question that they have chosen haploid strain. So a heterozygous coming into action. Heterozygous means one will be the dominant allele. The mutant allele will be suppressed by the dominant allele. So whether YFG1 is nonsense mutation or whatever mutation, the dominant allele will suppress that mutation and the dominant allele will produce the normal phenotype and the cell will be able to grow at 42 degrees Celsius. That is in case of heterozygous. But here in the question they have told it is haploid strain. So YFG1 will result, if it is nonsense mutation, will result in the production of non-functional proteins. And non-functional proteins or null alleles, they will be non-functional at all temperatures. Okay, null allele, they will be non-functional at all temperatures. But they have mentioned that YFG1 is having impaired cell growth at 42 degrees Celsius. That doesn't mean that they will not grow at lower temperatures. They are growing at lower temperatures. Only thing they are not growing at 42 degrees Celsius. Okay. They have become temperature sensitive. That's what I told. Okay. Earlier when I was discussing the question. So, because it is a nonsense mutation, null allele is produced, and because of the null allele, they will result in cells which are non-functional at all temperatures. So here they have shown it is temperature sensitive mutation, or they are showing the temperature sensitive phenotype. So YFG1 need not be a nonsense mutation. It could be any mutation other than nonsense mutation, like it can be frame shift or it can be missense or it can be any other, but not nonsense mutation. So as per option D, if SUP1 is a nonsense suppressor, so I told you YFG1 has to be a nonsense mutation if SUP1 has to be a nonsense suppressor. Now YFG1 is not a nonsense mutation, so SUP1 being a nonsense suppressor is not correct. Okay, so that makes option 3 as the right answer and D wrong. Now next one. Two closely related individuals suffering from a congenital disease, that is by birth they are having the disease, have several children. A genetic counselor construct the following pedigree of the family and conclude that disease is caused by mutations in at least 2 genes. Okay, they have mentioned 2 genes, so we will take into consideration the dihybrid case. So how many offsprings are there? 16 offsprings are there. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 are there, and how many are deceased? 3 and 3, 6 and 9. So 9 are deceased. Okay. The others are not deceased. So 2 genes we take dihybrid condition into consideration. Now further, based on this information, the following statements were made. At least one of the children will not carry any mutation linked to the condition. Okay, wait. We will come to that later. The mutation causing this disease are recessive. If this is in the recessive condition, that is if it is having this genotype, that is both the parents are having small a, small b, then all the offsprings will have small a, small b. Okay. Then all the offsprings will have small a, small b, because none of the parents are having any dominant allele in them to supply to the offsprings. So here you can see there are non-diseased offsprings also. So this mutation is being caused not by a recessive gene. So B is wrong. At least one of the mutant gene is on the Y chromosome. If that was the case, what about these males who are unaffected? They should also have been affected by this if it was in the Y chromosome. See, the females all are XX. How did they get the Y gene? Not possible. So, That is also not correct. Simultaneous heterozygous mutations in the involved gene can cause the disease. Yes, that could be. Now if you see here, capital A, capital B. Okay. If these two are mutated, that is, it is a dominant mutation, then you will get 9 offsprings as in dihybrid cross, which are carrying the disease. That means simultaneous heterozygous mutations. Heterozygous here means in different alleles. So A is one allele, B is one allele. So these two are different alleles. They both are mutated, then you will get mutated children. But if only one is mutated, see here, in this case only A is mutated, in this case only B is mutated, you will not get the diseased condition. That is why you got some offsprings which are not diseased. So that case D is correct. What about A? At least one of the children will not carry any mutation linked to the condition. Yes, see here, this case, small a, small b, only one offspring. Yes, one of the children will not carry any mutations. So that's correct. Okay. So that makes option A and D as your answer. Then next one. A mutation in a plant gene is female gametophyte lethal. That means it will affect the viability or function of the female gametophyte, that is the embryo sac in plants. That is what is meant by female gametophyte lethal. And embryo sac is mutated means it will affect the fertilization, seed development, etc. in plant. So this mutant allele can be normally transmitted through pollen. Okay, now this mutant allele, it is transmitted only through pollen but not through egg. What will happen if it is transmitted through egg? The embryos abort. Okay, resulting in inviable seeds irrespective of the male allele. So if the male allele is supplying the normal gene and the female allele is supplying the defective or gametophyte lethal allele, whether the male allele is normal, still then the female allele will be more active and it will result in the formation of inviable seeds irrespective of the male allele. So a student harvested seeds from a heterozygous mutant plant and grew a total of 100 plants. Okay, what is the expected number of homozygous plants in the population? Now you see that in heterozygous plant, so this is the male parent and which is heterozygous mutant plant, and this is the female parent which is again a heterozygous mutant plant. So the student harvested seeds from this and the seeds will produce a genotype. What is that? If it is male parent, it will produce the capital A genotype and the small a genotype. Now small a, we will take that it is the one which is causing the lethal mutation. Now the female parent, it will not produce the small a gamete. Why? Because if it is transmitted through eggs, the embryos will abort. So it will not produce that gamete. Okay, but the mutant allele can be transmitted through the pollen. They have mentioned in the question. This mutant allele can normally be transmitted through pollen. So pollen will produce both. Now upon crossing you will get capital A which is 50 and capital A small a which is 50. They have asked expected number of homozygous plant, so it is 50. So the answer here is option 2, 50. Now in a sesame the seed coat color is of two types, brown and white. True breeding white and brown seeded plants were reciprocally crossed and the results are given below. Reciprocal means if in the first cross you take white as the female parent, in the reciprocal cross white will become the male parent. That is meant by reciprocal cross. You change the sex of the parents, okay, and cross them. So which one of the following type of inheritance explains the depicted transmission of seed color in sesame? Whether it is a plasmid, mitochondrial, cytoplasmic or nuclear mediated maternal effect. I will tell you what is it, both plastid and maternal effect. Now we will take the example of the given in the question itself. Now maternal inheritance means the characteristics or the traits are exclusively got from the mother to the offspring. That is the mother's mitochondria, the mitochondria will be obtained from the cytoplasm of the egg, whereas the father's sperm doesn't contribute any mitochondria to the offspring. Okay, so the cytoplasm will be always of maternal nature, not paternal. So based on the cytoplasm, the phenotype of the offsprings will be determined. Okay, that is in case of maternal inheritance. See here, it is the parent is, mother is white, father is brown. You will get F1 which is white because even though it is a recessive gene and father is a dominant, you will get the children white because the mother is supplying the mitochondria, not the father, so white. Okay. Now if you do an F2 progeny, again the mother is supplying the mitochondria, father is not supplying the mitochondria. So if you cross these two or self the F1, BB and a capital B small b, you will get all offspring as white irrespective of their genotype. See here, all offspring will be white because mother is the supplier of the mitochondria. That is maternal inheritance. Now what is the case if it is maternal effect? Now maternal effect, the phenotype of the offspring is determined by the genotype of the mother, and the offspring's genotype doesn't have any role here. That is maternal effect. Okay, first is inheritance, inherited. Mitochondria inherited from the mother. Maternal effect means maternal genotype is showing its effect on the offspring. Okay, that is maternal effect. Okay, now see this case. So here again white, brown. What do you get? F1, genotype is capital B small b, it has to be brown as per Mendelian inheritance. But what do you see in the phenotype? It is white. Why? Because maternal genotype is white. Small b, it is coding for the white protein. So the phenotype will be white. Now if you self these, that is phenotype is white and genotype is for brown, you take the male and the female parent. And what do you see? You get the phenotype brown. Why? Not white. If it was maternal inheritance, it would have been white as in this case. But here it is brown. Why? Because the maternal genotype here it is coding for the brown protein. So the phenotype of the offsprings, whether it is small b also, it will be brown. That is maternal effect. Now coming to the question. So here the white female is crossed with the brown male and you get 100% white. Happy. Why? You will think it is cytoplasmic inheritance. But if you see the F2 generation, you are not getting white as in the case of cytoplasmic or maternal inheritance. You are getting brown. So it is maternal effect, not cytoplasmic inheritance. So your answer is option 2, nuclear gene mediated maternal effect. Then coming to next one, based on homology, a protein CG2024 function as a heterotetramer. Okay. Homotetramer. Okay, so all these are of the same domains. Okay. The function of each unit within the tetramer is essential for its catalytic activity. So CG2024 protein has 3 domains. Okay, 1, 2, 3. The fourth domain they have not mentioned anything. Domain A is essential for tetramerization. So if this is domain A, tetramerization has to be there. Domain B is essential for catalytic activity. This is for catalytic activity, and C, it doesn't contribute to the function at all. So whether C is mutated, C is not there, no harm, the protein is going to function. So 3 mutations A star, B star, C star in the ABC domain of CG 2024 respectively have been identified. The A star, B star disrupt the function of their respective domain. Why? Because A is essential for tetramerization. Without tetramerization, the protein is not going to function. B is essential for catalytic activity. Again, no catalytic activity. What is the function of it being a protein or an enzyme? No function. So it is also disruption of the function. But domain C, I told you, it doesn't contribute to the function at all. What will happen if it is mutated? No change in the function of the enzyme or the protein. Okay, so based on this information, which of the following option correctly describe the nature of mutations of A star, B star, C star in the same order? So A and B, I will tell you, they are 100% dominant mutations. Okay, but C, it is not recessive. It is called as amorphic mutation. Okay, amorphic is null mutation. Okay, amorphic means null mutation that result in the complete loss of function of the gene. Okay, and there will be complete loss of protein's normal function also in case of amorphic mutation, as if the gene is not present there at all. That is amorphic mutation. Now C doesn't contribute to the function at all. So whether it is mutated, no harm. So it is a case of amorphic mutation, as if the gene is not there at all or the gene is entirely absent. So it is amorphic mutation. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of CSIR been discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, C for CSIR net exams, and there you will find. Thank you.